Today I will discuss about the Rhizopus fungi. The systematic position is kingdom Mycoda, division Eumycoda, subdivision Zygomycotina, class Zygomycetes, order Mucorales, family Muceraceae, genus Rhizopus. There are some Indian species Rhizopus stolonifer and Rhizopus oryza. This fungi commonly called as bread mold, black mold, pin mold. It is saprophyte type, grow upon the jam, jelly, vegetable, fruit, etc. It is white cottony outgrowth like structure. During maturation, reproductive organ became into black color or green color, later into black, finally black color. Some species Rhizopus articarp, it is a weak parasite. In sweet potato, it causes soft rot disease, mycelia type, whitey cottony, stolon, accepted body. Those fungi are mycelia type and whitey cottony like structure and stolon appear and accepted body. Vegetative body. Vegetative body. Mycelium is long, slender, much branched hyphae. It is cynocytic, that is, the whole body is accepted. Finally, septa form during the reproductive organ produce. Stolon appear. This fungi is cynocytic, but when the reproductive organ produce, then the septal formation is found and the rhizoidal hyphae is present. This is the image of the Rhizopus. This is the rhizoidal hyphae and this is the mycelia and stolon, and this outgrowth upper outwardly structure sporangia. This is the vegetative body structure. It is accepted hyphae, multinucleate, vacuole are found and possess well drop glycogen as a reserve food material, and nucleus present, and the cell wall made of protein, fat, magnesium, chitin and calcium salt. In this cell there are nucleus, endoplasmic reticulum, ribosome, mitochondria, vacuole are present. Reproduction, mainly two types of reproduction are found: asexual and sexual reproduction. Asexual reproduction by the sporangiophore and chlamydospore, and sexual reproduction by the chamaedogeal copulation. Asexual reproduction, when the mycelia mature and the next outgrowth like structure are formed. This outgrowth like structure upwardly grow and finally the protoplasm of the mycelia upwardly grow and dense in the upper portion and form the sporangiophore, and at the tip portion it is the sporangium. Here the cytoplasm or protoplasm material upwardly go and aggregate and solen and form the sporangia. A protoplasm remain into two states, and the protoplasm densely occur here and the central lightly present protoplasm, and between the middle or between the two parts here a cleavage cavity are found which separate the dense and light protoplasm. So here we found the protoplasm remain into dense protoplasm at the side portion and at the central light protoplasm and separated by cleavage cavity. Next, the dense sporoplasm produce multinucleate zoosporangium, and the inner, it is called columella, which is hygroscopic, water absorbs, and the cleavage cavity this become as a vacuole which different two part, inner and outer. Inner portion called columella, outer portion is called sporoplasm. The dense protoplasm here contain nucleus, and each nucleus contain the protoplasm and produce many sporangiospore. The sporangiospore release when the columella absorbs the water and release the spore in the outside. Next, the sporangium germinated and produce finally mycelia. Again mycelia produce outgrowth. This life cycle may be continuous. This is the sexual reproduction by sporangiophore, and another is by the chlamydospore. Chlamydospore, when the unfavorable condition came, then the protoplasm or rhizophore material aggregated and a thick wall like structure is formed and septate wall formed. Here that cytoplasm, protoplasm or rhizophore material are contained. Next, this containing gradient represent as chlamydospore. It is very thick walled. And next, chlamydospore in favorable condition germinated and produce hyphae germinated and produce hyphae. This is the actual life cycle by chlamydospore. Next, I will discuss about the sexual reproduction. The sexual reproduction takes place by the opposite stain. One is plus and another is minus stain or hyphae. This plus and minus stain hyphae come to contact. When they come to contact near, then outgrowth like structure is produced. This is called progametangia. This is minus and this is plus hyphae. Now this outgrowth like structure, which is progametangia, they come to contact and a suspension like structure is formed. The protoplasm of minus and plus hyphae, here conjugation takes place. Here the protoplasm conjugation takes place. Next, we found that this mycelium a suspension is found where here the two cell protoplasm fusion take place and this solen like structure is formed. This is called suspensor. The suspensor next from the young zygospore, young zygospore, and young zygospore is thick walled and spiny into the structure. Zygospore are present. Next, here we found that the two layer is found as a of the spore, exospore and endospore. Exospore and endospore. The exospore two layered and endospore three layered. Next, here the germ sporangium are initiate. and here the meiosis take place and meiospore are formed and meiospore released. Those meiospore germinate and and finally produce hyphae plus minus hyphae. So rhizopas reproduce reproduced by sexual and asexual. Asexual reproduction takes place by the sporangiospore and the chlamydospore, and sexual reproduction by the gametangel copulation. Here plus and minus type of hyphae gametangel copulation is take place, and such this process sexual reproduction is happen. And this is the sexual reproduction by by sporangiospore and chlamydospore, and this is the two process of asexual reproduction, and this is the sexual reproduction. Thank you.